Log on, tune in, find out. Another good idea from Cambridge.
Our keynote speaker for the afternoon is Dr. Holland, who is from Tasmania, even though on somewhere it says something else, but don't believe that. And she's going to be talking about making the most of DART data.
Thank you, Dan. So I think the microphone is on. Can you hear me at the back all right? Very good. Yeah, so up until a year ago I was at Massey University, which is what my name tag still says, but yeah, moved to the University of Tasmania about a year ago, and one of the people that I started talking to was a plant scientist by the name of Dot Steen, and she had what I've sort of come to discover is a really fascinating data set, and that's like a new type of data that I hadn't come across before in my sort of travels in the land of phylogenetics. So you notice there's a couple of other names here on the title slide. Michael Woodham's, his name is in red because almost all the results I'm discussing are actually his work. He's doing a postdoc with us at the moment. The other name is Vince, who I've been pestering for the last week in Norwich, and he's also sort of been a collaborator on this project. So do all those in advance. Okay, so DART data. So DART is an acronym, stands for Diversity Array Technologies, which is particularly useless because it doesn't actually tell you any more really about what they might be. But this is my sort of understanding of how these phylogenetic markers are created. So start off with a genome, indicated here by this line. So that could be just from one species, or it could be from a small set of species. And you've got two enzymes, one that recognises a 6 base pair sequence, one that recognises a 4 base pair sequence. And so what you do is you chop up your DNA with this, so we call one the rare cutter just because it happens only 1/16 of the time that the other one will come up, and one frequent cutter. So we chop up our DNA everywhere we see one of those recognition sites. But then the rare cutters, which I've sort of symbolised with these blue boxes, are somehow special, that they have some adapters on the end of them that are then recognised, and so you can use the magic of PCR to get multiple copies of just these fragments. So basically, if you have a fragment that has two of these rare ends, it will be amplified, retained. If you have a fragment that say is between two of these green cutters, you'll lose that, it will disappear. Likewise, if you have one with two different kinds of ends, you won't get that either. And so what that basically does is give you a pretty random subset of your genome. And so the sort of data that Dot has is for eucalyptus trees, and in her case she gets about 8,500 of these fragments. And they can be anywhere from 100 base pairs to maybe sort of 1,000 or so base pairs long. So once you've got this, what's called the genomic representation, so this random snapshot of your genome, you somehow stick it to a microarray, and you can then analyse other samples by simply digesting them with those same two enzymes, running it across that microarray, and if you get a match somewhere, you get some colour dye, and if you don't have a match, you get a different colour. So you're basically getting presence. You can measure presence or absence of each of these fragments. So a lot of you might be familiar with AFLP or RFLP, so various fragment length-based techniques. This is a bit like that, but it is nicer in that with AFLP, if you have two fragments of the same size, like they'll run to the same point in the gel, you'll call them both a 1. They might not actually be the same bit of DNA, they might just have the same length. Here, because you're doing DNA hybridization, you're really quite sure if you get a joint presence, so that you're looking at homologous bits of DNA. You're not looking at something that's just accidentally the same size. So they're sort of a little bit like a SNP chip, but they're also a little bit like AFLP. And so got a little disclaimer down the bottom here. So fragments are either present or absent. In reality, you also will get some question marks, so sometimes it will be hard to make a call. So let's sort of boil down what properties does this sort of data have. So it's pretty much binary if we ignore the question marks for now. So you've just got zeros and ones, presence or absence. It's randomly scattered across the genome. So that's perhaps quite nice in that you might sort of avoid, so we know that there are dangers in just looking at a single gene and then trying to infer something about species phylogeny, because a single gene or location might not match the history of a whole genome. So it's nice that you've got this random scattering. And the other thing is, these fragments are typically quite long, so minimum size would be about, I think, 100 base pairs or so. So you're really quite unlikely to get a match if it's not really homologous bits of DNA. So you're pretty unlikely to just by random chance get two 100 base pair fragments that match each other. I mean, you might have there's always weird things that really go on in biology, like you might have repeat regions and you might not be. But as a first mathematical approximation, we can sort of say that if we end up with two ones, so two presences, that they're truly homologous. However, it's not quite like this very nice data that Dan was discussing, like your sort of infinite sites type model, because you can have multiple losses. So you're not saying that every single character like this will be some exact match on a tree or be convex on a tree. Because though you can only probably think of gaining a particular character once somewhere on the tree, you could lose it at multiple positions over the tree. So the other weird thing about this data that's a little bit different from other sorts of data sets I've worked with in the past is this ascertainment bias or censoring effect. I'm not quite sure what to call it, but the fact that you only will see fragments if they appear in the reference genomes that you use to make that microchip in the first place. So in the case of the eucalyptus data, these chips weren't made because people thought it would be a wonderful way to do phylogeny. They were made for some sort of economic purposes and trying to find, I think, sort of quantitative trait loci and things like that. So the reference set is basically the 6 or 7 most economically important eucalypt trees, not some nice sort of sample from throughout your tree. So how should we model, what should our mathematical model of this sort of data be like? So basically it's a stochastic Dollo process. So Dollo meaning you can have a single gain and multiple losses. Stochastic meaning that if you have a long edge in your tree, you're going to be more likely to lose a fragment down it than if you only have a short edge. So this Dollo bit means basically that parallel gains are forbidden. We're going to assume that all our fragments are lost at some constant rate, and that's a sort of memoryless process, so your chance of losing a fragment only sort of depends on your current state of your DNA. It doesn't depend on any of the more ancient past. This is a wee bit of a simplification to assume that all fragments are lost at the same rate. Like if we think about, and then we go back to this first picture here. So you can imagine, so how might you lose a fragment? So if we have a fragment here, one way that you would kill that fragment is if you had a mutation in one of these recognition sites, of which there are obviously 2, one at each end. And so that isn't going to depend at all on the length of the fragment. But the other way that you could lose something is if you had a mutation in here that somehow made one of these other recognition sites. So you're actually slightly more likely, you've got a slightly higher death rate if you're a long fragment than if you're a short fragment. But in some sort of simulations that we looked at, it seemed like that effect was reasonably slight. So the biggest way that the dominant sort of term when you're thinking about the death of these fragments is mutations that hit one of these recognition sites. So our modelling assumption is basically that all the fragments are lost at the same rate. So that's a slight simplification of reality. And then you've just got some basically exponential sort of decay type process. Okay, so just a little picture just to sort of summarise the situation that we've got. So I'm calling it basically a censored Dollo process. So the censoring coming from the fact you won't see something unless it's in your reference genome. So say in this picture here that the square is our reference genome, so that's the one we've used to make our microchip. So if you have some fragment, you know, that happens to have two of these rare ends in this genome over here, but it wasn't over here, you just won't see it. It just won't turn up on your chip. So any pattern where this is, you know, a 0 at the reference taxon is just unobservable. And then if you sort of say you have a pattern like this one here, so it's in the reference genome and in this sort of taxon on the left, and you've really, if you believe the Dollo model, you've only got one way you're allowed to explain that. You have to, because you're only allowed a single gain, can't have an independent gain here and here. You say, well, it must then sort of be at the ancestor of all the places where it's present, and then it's just been lost once down here. Okay, so like I said, this sort of data wasn't originally made to do phylogenetics, but nice cheap way to get, you know, 8,500 characters for 94 eucalypt species. So people have obviously started to think, well, let's use it for phylogenetics. And so there's been a couple of papers that have appeared, or people also would just do things like multi-dimensional scaling or MDS sort of type things on the distances. And the most common distance that people seem to have used in the literature is the Hamming distance, which of course is everyone's favourite distance because it's so easy to understand. But it turns out to be a shockingly poor choice for this kind of data. So this is just a sort of cartoon example to convince you of that if you need convincing. So here's our reference taxon. We've got 16 fragments that are present there. And we're assuming on this tree that down each branch there's a 50-50 chance that a fragment survives or it's lost. So if we come down this branch here, we've lost half of our fragments and retained half. Down here we lose another half, lose another half going this way, another half there, and another half to there. So the true distances on this tree, the distance between ref and B should be exactly the same as the distance between C and D. In terms of actual length of time or probabilities lost, they should be the same. But when you look at the Hamming distance here, you get a distance of 3/4, whereas the Hamming distance between C and D is tiny, the 2/16. So the problem is that your Hamming distance sort of treats these shared absences exactly the same as a shared presence, which is not really what you want to do with a Dollo model. And so the effect of that on tree reconstruction is you shrink. So here A was the reference taxon. You can see you basically shrink all the edges as you get further away from your reference taxon. Okay, so we wanted to come up with a distance correction that would be appropriate for this kind of data. So it would capture the fact that it's a Dollo process and also capture that we've got this ascertainment bias that we only see things that are in our reference. And so let's start with the simplest case possible. We've got R, a reference taxon. We're trying to measure this distance between A and B. So what data do we have available? So basically, for the purpose of this, all the distance corrections that we're going to look at, there's really only sort of 4 quantities that are of any significance. So we've got the number of fragments that are absent at both A and B. Then we count the ones that are absent at A, present at B, and so on. So we've got these sort of 4 possibilities. So then the Hamming distance that we used before would simply be, say, N01 plus N10 divided by the sum of all of those possibilities. But we're going to want to do something a bit different than that. So Michael and Vince and I were sort of starting to toss around some ideas about this back in February, and Michael had the following simple but key observation, which is that if we just consider fragments that are present, say at taxon A, and we know they're also present at the reference taxon because that's just by definition of this kind of data. which means, and because we've got a Dollo process, we can't have parallel gains. They must be present everywhere along the path between R and A. In particular, they are present at that internal node there, X. So what we can basically get at then, we just sort of look first in the very middle of this slide here, we've got basically a proportion of fragments that survive down this edge here. So we've got ones, the N11 fragments, so they're present here, and we're just dividing by the total number that are present at A. So this term in the square bracket here is just the proportion of fragments that survive from X to B. And so we're saying in our model that we've got this sort of exponential rate at which fragments are being lost, so some kind of decay process. So we can basically then, to recover this R times T, we want to take the log of this proportion here, and well, minus the log. And that's going to give us our distance from X to B. And we then just sort of reverse the argument and consider only fragments that are present at B, what proportion of them get lost down here, to get our distance correction. So it's a little bit odd looking compared to, say, a Hamming distance or a Jacquard distance, but it seems to follow reasonably logically from the properties of this kind of data. But of course, we don't really want to just compare to Hamming. That would be really setting up a straw man. We want to compare to some of the other distances that people have used and other distances that we think might be more appropriate. So, been a couple of papers where people have used log det corrections on this kind of data. Also the Jacquard, found people who'd used that. Then down here we've got some that we thought might do a bit better. So HS stands for Hewson and Steele. They had a paper in 2004 that was looking at gene content data and how you might correct distances for them. So think about the gene content situation. It's almost exactly the same as our situation, that it's stochastic Dollo, but you don't have any problem really with an ascertainment bias. So it's sort of, and so this distance here, you needed to know what was the average number of genes in a genome. And so we thought, well, we don't quite know that, but for any two genomes we're comparing, we can say what's the average number of fragments. And if you make that simplification, then so this isn't quite the distance that's in the 2004 paper, but it's what it boils down to if you make that simplifying assumption. And there's also distances that have been developed to deal with restriction fragment type data. So looked at those as well. And so basically we wanted to test how well this distance correction seemed to perform in practice. So do what sort of maths geeks and computer geeks love to do. We simulate ourselves some data. So two scenarios really. One where we enforce a clock, and one where we don't, where we basically will sort of simulate a Yule tree but then reassign branch lengths, just picking from a uniform distribution between, say, 0.05 and 0.4. Where this is, I'm never quite, Mike always parameterizes things in a slightly different way than I expect, but I believe that's the probability of a loss down an edge. Seems like the most logical thing for it to be. So found another very useful fact in the 2004 paper of Hewson and Steele, which said it doesn't matter where you put the root in a Dollo process. At least I hope that's what we thought it said. And so we figure this is great. We can just start from an arbitrary root, and we'll start with, say, if you think the mean number of fragments in any genome is m. You can start with a sort of Poisson number of fragments with mean m. And then those fragments we start with can only be lost as we sort of simulate down the tree, but on any new edges you might gain some new fragments that you haven't seen before. So we just sort of simulate our way down. Okay, so I was wrong about the parameterization, but I have it here. So the edge length is L, probability a given fragment survives that edge is exponential minus L. And then we'll have some new number of fragments. And so what this sort of procedure does is it means that your mean number of fragments anywhere you look in the tree is a constant n. And so the other sort of crucial thing when you're simulating this data was to get that ascertainment bias in. So we start off by ignoring that. We just do a stochastic Dollo simulation. But then we arbitrarily, so we pick at random one or sometimes two of our genomes to be the reference genomes. And then we'll throw away any data we've simulated that isn't present in at least one of our reference genomes. And when we tried sort of a few different scenarios, ones where we have reference taxa that are then included in the final set we want to work with, ones where we pick a reference taxon, use it to mask out fragments we wouldn't see, but then throw the references away. And we also just tried the scenario where all of your taxa are in the reference set, so basically no ascertainment bias. So if you get the feeling that I'm hiding something, I am, but it will be revealed a little bit later. Okay, so what are we looking at here? So along the bottom here we've got our different distance metrics. So LD is log det, H is Hamming, J for Jacquard, NL's the restriction site-based one, Houston Steel, log of a Jacquard distance, and our new one, the DART distance. No, we haven't ever found anyone who's taken the log of a Jacquard distance before either. Michael decided that would be a fun thing to do. It actually turns out to work quite well. And then we measure a root mean square deviation from true. So how well do our distances match the known distances that we generated on, and we sort of increase our number of fragments. So of course what we hope is that as this number gets bigger, we're getting close to 0 here. So you can see log det does appallingly badly. Hamming and Jacquard also both do badly. And these four here all do reasonably well, really, by comparison. So that was the case where all taxa were references, so that's sort of without any ascertainment bias. If we look now at the case where you have a reference taxon and then throw it away, you can see DART distances are doing pretty well. If you have a reference taxon and then keep it in your data set, DART distances are still doing pretty well. We're not quite sure why that makes such a big difference to our result, but we've still got this same observation that log det and Hamming are sort of poor ways to treat this data. So that's just looking at how well the distance matrices match. And the other thing is, well, if you then build a tree on those distances, do you get it right or not? So it's an obvious other thing to ask. So we make a distance matrix and then we just use fast ME as our method in all cases to do the tree reconstruction. I think Michael tried neighbour joining in UPGMA as well, but found this sort of always dominated in terms of accuracy. And so the different colours here represent your different scenarios of what you do with the reference taxa. So you have one reference taxa that's included in your data set, or one that's excluded, or two that are included, and so on. And this is percentage incorrect. So we want little bars. Little bars is good, big bars is bad. So you can see log det here, you really couldn't do much worse than it's wrong 100% of the time. So I think you'll notice on the front of your little folder that you got given from the Isaac Newton Institute that on the blackboard there is a little log det written there, so put a little black mark by that. It's not good on this kind of data. The DART distance does better than the others in these two cases here where there's just a single reference taxa. So that's the case it's designed for, so you'd hope that it did better than other distances there. I should say off to the right here is Dollo parsimony, so not a distance-based method, just a parsimony-based method, which also does pretty well. Should, I guess, expect it to. I mean, it doesn't have anything in there. So it's parsimony, so it ignores branch links, but it is at least it's got the right model in terms of the Dollo-ness. So we don't do as well in the case where you have multiple reference taxa. So that was sort of the next kind of challenge to think about. The other thing, so this is the clock-like. So the previous one was for the non-clock-like trees. If you have clock-like trees, you lose a lot of ability to tell differences between things. Everything all of these methods here are sort of much of a muchness. Log det and Hamming still a little bit worse. So, so the other thing Michael thought would be interesting to look at is are the different distance corrections sort of biased towards different tree shapes? So if you have a more or less balanced tree shape, does that affect your ability to reconstruct things? So what you're hoping really is, the log Jacquard's doing best here, and it has no effect at all of the number of cherries. So if you've got more cherries, roughly the more balanced your tree shape is. And so the restriction site-based distance is doing best on unbalanced trees, as is the Hewson steel, as is our new distance. Hamming and Jacquard have a sort of a bias in the reverse direction. Okay, so that's all good as far as it goes, but what about if you have more than one reference taxon? It's going to sort of make life sort of more interesting. So if we have again our sort of cartoony type example of fragments just being lost, again down each edge of this tree they were losing half. So if we just had R as our only reference taxon, so A, B and S are just normal taxa, we've made our chip with respect to R, then we'd only see the blue fragments. We'll only see the coloured fragments rather. We don't see any of the black ones. And then in that case we get a good sort of estimate of our distance between A and B. However, if we see any fragment that appears in either R or S, then what we end up doing, we just apply our sort of straightforward DART distances, we end up with an overestimate. Because, well, we'll get to because later, but anyway, this is just to convince you that there's a problem here, that we can't just take our simple correction and apply it to the case with multiple references. Okay, so what we want to do is to generalize to the scenario where we have multiple reference taxa. And so the first sort of conceptual thing was that you could think about, so you have this notation here, say you're trying to calculate the distance between A and B, conditioned on or with reference to some other genome R. So what I mean by that is I'm going to compute that same DART distance that I showed you before, but I'm only going to look at sites, or that should really say fragments, not sites, sorry, that are present at some genome R. So here, I mean, I've used R because we're treating it like a reference genome, but we're saying it may not actually be a reference genome. It's just some genome that we're using as an arbitrary reference point. And then, so this distance here then takes basically a weighted average of this distance evaluated with respect to some particular genome R, and just averages over all possible genomes R. So it's kind of basically the situation where someone gives you some of this data, you have no idea what the reference taxon is or if it's included or not. So in reality the situation is a bit better than that, like you pretty much do know typically what the reference taxon, what the reference taxa are. Like so for instance in the eucalypt data, I think there's 94 taxa, something like 7 or 8 of them are references, they're known. And it's also known for each particular fragment or each site in your character matrix which reference taxon it belongs to. So in that case you should be able to just partition up your character matrix into sort of different sets that are known to have come from different reference taxa, and just take some kind of sensible weighted average over those. So simulations to test that idea are sort of still ongoing. Poor Michael, I left him with my slightly evil head of department and said, you know, Michael should be left to work on these problems so I can talk to him, and he said, so I got an email from Michael about 2 days after I left saying, they've made me teach a regression paper and I've never taught it before in my life, so I won't be answering any of your emails for the next week while I hurriedly prepare. So I was hoping we might get some results here, but poor Michael has been afflicted with teaching while I get to jaunt around Europe. Sad. Okay, so this then is just going back to the results we looked at before and saying how does this, the generalized version work. So this is the one that sort of says, well, we'll pretend we don't know anything at all about what the reference taxon are, and we'll just condition on each possible reference in turn and then take an average. And so this is the situation here where really all of the taxa were reference taxa, and it goes reasonably well. So this is the fun picture. So in this case we have, so this is again showing how often you're getting the tree topology incorrect. So we want little bars, big bars are bad. So, and this is this new distance we've just proposed, this generalized DART. So this is weird. So it does incredibly well if you have the reference taxa included in the set that you consider, and it does incredibly badly if you have some reference taxa that you use to generate your data and then throw them out. So this is basically the point at which I get to Norwich last week and sort of say, Vince, this is really weird. This is, I think, the big thing that we should try and look at this week, try and understand what on earth is going on with this situation over here. So one of the obvious things is that this is just a 0-1 measure. You got the tree right, you got the tree wrong, but it might paint a very different picture if we look at percentage of splits correct. So like these trees here that are wrong, are they only wrong one split, or are they just, they're completely wrong? So that's something we have to go back and take a look at. I didn't quite, so this is sort of a bit random. This is the Eucalypt data. I thought didn't quite know where to fit it in with the rest of the talk, so it's just randomly appearing here. But this is just to convince you that that sort of dealing sensibly with multiple reference taxa is really an issue that we're going to have to get our head around. So this little histogram down the bottom here basically shows, it's just a frequency histogram of how many zeros there are in the character matrix for any particular row. So how many fragments are missing or absent in any particular row. So we've got some down here where, so this is out of about 8,500 characters, they have about 6,000, about 2,000 are missing. Then there's another mode over here where you've got about 3,000 missing, about 5,000 present, and then another mode down here. So it's really uneven. The reference taxa, like I said, were chosen just because of economic importance, and I think there are 6 of them in one clade of the tree, and another 2 sort of scattered somewhat more randomly. So you've got a weird distribution of zeros and ones throughout your character matrix depending on how close or far away you are from this main set of reference taxa. Dot was reasonably happy with the tree that we got using the generalized DART distance, but for obvious reasons with the simulation that I just showed you, don't really 100% trust this generalized distance here until we sort of understand what's going on with it a bit better. So wind back time a week and I arrive in Norwich and basically give this talk to people there, and this is sort of the one question that's most sort of plaguing us. What the heck is going on with this sort of generalized distance? So what I decided to do is basically just try and work through a set of simulations, which are wonderful. You don't really have to turn your brain on very much for simulations. I thought, let's work through some small example really thoroughly by hand and see if we can understand what's going on. So actually try and write down what are your probabilities of seeing different patterns under some model. And so the single reference taxon case again is nice because you can just root the tree at that reference taxon, and then you can completely forget about fragments being gained because you won't ever see them, right? So if you have a single reference taxon case, you really just have to consider all the different ways in which fragments can be lost down branches of that tree. So for example, if you see a pattern that's a 1 at the reference and it's also 1 at C and D, then you know for sure that it can't have been lost down either of those 2, well, it can't have been lost down any of these 3 edges here because it can't have been gained a second time. And so you sort of basically you've fixed that it must be present at each of these internal nodes here. And so you can write down kind of an easy expression for the probability of seeing that pattern. There are other ones where you have to sum over different terms. I've just sort of picked on a sort of a nicer one there. And just to remind you, so this, so here just sort of concentrating for now on trying to calculate the distance, say, between taxon A and taxon C. Just remind you of so this is our DART distance. This was that, you know, the formula that we came up with sort of based on the argument to do a single reference taxa. And you see it's got these terms in it, these fractions, so the number of sites that are present here in A, so absent in A, present in C, ones that are present in both, and so on. So these terms here are sort of obviously pretty crucial. So if we just pick a simple example where we set all these lost probabilities just equal to the same number, 0.01, so it's easy to see what's going on. And we have a look at these two fractional terms here that appear in our distance correction. So we can work out what our probabilities are of any particular pattern, so we can sort of marginalize these basically to get, well, what patterns give us this n, sum over the right ones to, so just basically get a probability of a 0, 1. I think there's a slight fudge factor here that we need to think about. I think if you haven't, probably many people in the audience know, but if you have the expectation of x over y, I'm not sure that that's exactly equal to the expectation of x over the expectation of y, but it's also thought it's probably not a bad first approximation. So all of these numbers are sort of with regard to that fudge factor that we probably need to work out exactly what bias that's introducing. But you can see what's going on in this little table here. We work out our distance between A and C, so we set all our probabilities 0.01, so the distance really should be 1 plus 1 plus 1 plus 1 is 0.04. So our DART distance is picking that up fine if we actually give it the real reference taxon R. But if, say, in our generalized DART distance, what we'd be saying is, well, we'll pretend we don't know what the reference taxon is, and we'll just take an average over this distance with regard, say, to R and B and D. You can see that these ones down here are underestimating that distance, and the reason for that is, so if I use the same argument, so let's think about using D as our kind of pretend reference taxon. So then the sort of the 3 taxon tree that we kind of care about with respect to that argument before is traced out here, right? And what we're hoping to be able to do is measure, you know, so using one sort of half of this formula here, we're trying to measure essentially this distance, and the other term we're trying to measure this distance here. But because we have this reference taxon that, you know, we don't know about, that we've forgotten about, to get something that's present at C, it actually must be at all of these internal nodes here. So essentially, by treating D as a reference taxon when it's really not one, we lose any ability to measure this distance in the middle here. So that sort of drops out. So what we're picking up is basically just this 0.02. So we've lost our ability to measure this middle part of the distance. We're just measuring this bit and this bit. So that was quite instructive, because it basically says, well, if you know what the reference taxa are, you better really ought to just average over those. If you don't know what the reference taxa are, you're actually probably better to take a maximum than to take an average. How am I going? Not too bad. So another sort of example here, thinking about the case where you have 2 reference taxa, R and S. Again, so you might think, what's the probability? I want to be able to write down probabilities of seeing any particular pattern. So up to a fudge factor that I'll explain later, let's think about what's the probability that you see a fragment at S and at C. And here it gets, as soon as you have multiple reference taxa, life becomes more painful for calculating these probabilities, because you have to worry about gain as well as loss. So, so how could we see a pattern like that one there? So basically what could, this first term here relates to the probability that you actually have a gain on this edge here. So you have some fragment that's born up here and that survives this edge, and these two here that dies here. The next term is basically fragments that are born here, they die here, and they survive down these two edges. This term here is for fragments that are born here, and the last term is for fragments that are born here. You say, well, what do you mean? Weren't these p's supposed to be the probability of loss? What are you talking about? You're confusing me. So they come from the fact that in order to have this, constant number of fragments, so your probability of losing a fragment down any particular edge has to be the same as your probability of gaining a new one. So these here aren't really probabilities of loss anymore, these ones in blue. They're the probability of picking up a new fragment on that edge. If you want to have a process that's sort of balanced, that you have the same number of expected fragments at all points in the tree. Now this becomes sort of problematical when if you write down your probability of all of your patterns in this way, suddenly they don't sum to 1 anymore because of the fact that you don't really have, say if you had m fragments at every particular genome, that was your sort of equilibrium number, you actually end up seeing more fragments than that because you see ones that are present at R, ones that are present, or ones that are present at S. So actually your total number of fragments is going to be bigger than your mean fragment size. So you need to renormalize these, but that I haven't worried about because everything we do is just involving ratios of them anyway, so I figure that this normalization thing should come out in the wash. But this is, like I say, I have a different philosophy on talks than most of the other people, that I like to talk about things that are half done as opposed to things that are already published. So you should be aware that some of these sort of quick and dirty approaches may have to change in the final analysis. So this is just showing this this problem here. So if you consider then your probabilities of seeing, so this should really be P rather than N, so your probability of seeing any of those different patterns. If you look just with respect to reference taxon R, you get things nicely summing to 1. If you look just with respect to S, you get things nicely summing to 1. But if you actually consider all of them, then you get your probabilities no longer actually being probabilities unless you actually do this. So you need to renormalize these for them to actually be probabilities. And you can see then, so if we're trying to estimate this distance AB, if we just look with respect to R, we do fine. If we just look with respect to S, we do fine. But if we look at all fragments that are present in R or S, then we overestimate that distance. So those sort of hand kind of worked examples basically suggest that we need to go back and do some more simulations. So in the case of unknown, we're sort of interested in this case where maybe you don't know what the reference taxa are, then we want to think about the idea of taking a maximum over these distances with regard to all the possible reference taxa. And if you know what the references are, you probably don't want to be doing what we were doing with this generalized Dart where you average over everything. You really probably just want to average over the reference taxa and just treat them. And that sort of seems slightly nasty in the sense that if you have a fragment that's present in many of the reference taxa, it will appear in many of these sums. So it will sort of somehow have more influence than if you have a character that's only present in one or a small number of the reference taxa. So, okay. So that's sort of to date what we've done. Now we get into speculation, or even more speculation you might say. But so one of the things that we're really interested in looking at with this sort of data eventually is to what extent has hybridization sort of played a role with the eucalypt taxa. So I mean there's sort of theories that it might possibly be really quite a lot. So if you have a taxon, so if you're under a Dollo model, if you see some characters present, on one half of your tree and also on some other half, then you've really it's got to be present basically at the most recent common ancestor of all of the taxa where it's present. So it sort of gets forced right back to the root, and then you have to have potentially like a large number of, well here too, but you could potentially imagine if this was a more caterpillar shaped tree, you might really have a very large number of losses required to explain that. If you allow a borrowing or a hybridization, so you could have a gain here and then that fragment manages, due to say a hybridization event, end up over here, then you might be able to potentially explain some of these characters much more cheaply. So that started to remind me of talks I'd heard by people like Tal Dargan and Phil Martin and Jeff Nichols and Russell Gray, because the stochastic Dollo situation is certainly by no means unique to the DART data. In fact, sort of seems to actually turn up all over the place if you have basically complex traits in a situation where you know the one gain, multiple losses seems reasonable. So obviously gene content evolution, perhaps with horizontal gene transfer, or language evolution where you've got borrowing of words. They all seem like situations where we might be able to steal tools that have already been developed. In particular, I was interested by a talk that Tal gave, I think, 2 New Zealand phylogenetic meetings ago, and one that Russell gave 1 New Zealand phylogenetics meeting ago, where they're looking at the idea of minimum lateral networks. And so the idea behind this is, well, I call it, I don't know what they call it, I call it ancestral genome obesity, which is the idea of you can only lose things, and then you start to think about what has to be, so thinking in the gene content scenario, if you only allowed loss, then whenever you have a gene that appears on different parts of the tree, it must be also right at the root of the tree. So if you believe a Dollo model with no borrowing, then that tends to imply that your ancestral genome sizes have to be just absolutely absurdly huge. So the idea that these guys had was that, well, what if for every character you allow it to be borrowed once somewhere on the tree? What ancestral genome sizes do you infer then? What if you allow every character to be borrowed twice? Then what happens to ancestral genome sizes? So they're trying to basically have the requirement that your genome size stays the same across the tree, and then say, well, if we enforce that, how much borrowing does it imply? And then they make these quite neat looking networks. And so Russell had flashed up one like this for language evolution at the last New Zealand Phylogenetics meeting, and I thought, I want one of those for this data as well. And so I was in Dusseldorf a couple of weeks ago, and these are 2 of Bill and Tal's PhD students, Torsten and Shiju, and they very kindly massaged my data into the format that Tal's program expects, which is all to do with SQL databases about what genes do what, and so completely. You basically had to fake a whole lot of data for this program to work because it's expecting this gene content setting. So that was kind of amusing. But and then, so these are all the eucalypt taxa, and then it sort of basically this is a model where you're just allowing each character to be borrowed only once and then mapping on where that borrowing occurs. And you've got a funny artifact going on around the root. But this is kind of fun. I don't think we can take too much away from this at the moment, because obviously this is a really different situation than the one they're dealing with, where they're basically assuming, in terms of this setting, the same number of fragments all across the tree. That's what they're using to get these things, whereas we know for sure that because of our ascertainment bias, we don't have that. So, but it's certainly an area we want to try and think, can we borrow some of the tools they've developed in that work and apply it to this kind of data. And my last slide is an advertisement. Having a meeting in November where it will be cold and dark here in Europe, but sunny and warm in the southern hemisphere, right? And basically it's just for 2 days, but you could do some hiking in the middle and then go to the New Zealand Phylogenetics meeting in February. It'll be fun. Yeah, so it's just a couple of days, but we obviously would love, Jeremy and I would love to have people come for a longer visit if they were interested as well. Aimed more at the math sort of theoretical end of things, so you can read Jeremy's wonderful blurb that he's put up on the website here about all the fantastic different kinds of math that turn up in phylogenetics. And if you're keen, talk to either Jeremy or myself. I'd like to take the opportunity to really thank the organisers and invite questions.
There's certainly time for questions.
the actual structure of the tree that you produced, the eucalypt tree,
Yep.
anything that struck you about it that might give insight as to what's going on? So like the pen edges seem to be really long in it, but on the other hand, it seems to be pretty
So
clock-like in a sense.
it almost, like this plate here seems a bit shrunk with respect to the rest. So I haven't got the information about where the reference taxa are, but if I had to guess, I'd say there's a bunch of them probably down here, and that we're still possibly getting some kind of shrinkage effect. Because I think the distance correction we've used here, I think none of them are, like with regard to having, sat and thought a bit more about it last week, I don't think that any of them are quite right for the multiple reference taxa settings. I want to go back and I think just take, just treat each of the 8 known reference taxa in turn, get a distance based on each of them, and then average, and then see if that makes a difference to this tree. This was already thought to be a bit of an improvement by DOT over the Dollo parsimony tree for the same data, but I couldn't tell you why or even read the labels because 94 taxa is a lot. But she certainly thought it was a plausible tree. The back.
Yeah, so what's the problem with the Dollo parsimony? It seems like it performed best even in
So it certainly goes well. I mean, one of the problems might be if you had a real mixture of short and long branches, and that it would assume the same probability of a loss down any edge regardless of its length. So like I imagine if you simulated Dollo data on something like your Felsenstein zone tree, then Dollo parsimony might get into trouble. There's stochastic Dollo implemented as well. I've also tried, so Jeff Nichols has this program Trait Lab that runs in MATLAB, and it implements a sort of the maximum likelihood version of Dollo, or Bayesian sort of version. So I've sort of set that running for I think about 10 days, and my little furry caterpillar of convergence wasn't converging at the stage, so it was sort of a shame. But hence, I think that's part of the reason why we're sort of looking at distances to some extent. But I also had an interesting chat with Mark Succardo. I think in Beast has a stochastic Dollo model. Maybe Alexei can nod if that's true. Yes, that's true. So that would be another thing to try, especially if there was some way to actually tell it not to expect the patterns that you can't observe. I think it would be nice to be able to do it under a sort of a more likelihood framework as well. But we're sort of concentrating on distances at the moment for sort of reasons of, I guess, expediency in them. Another question? Yes.
Just for information. So the reason this was done, is it cheap? I mean,
very cheap. Yeah.
Really cheap. Is that
it's really cheap. Once you get your chip, it's really, really cheap.
It's good for very large clays, I guess.
Yeah, so like it's not at all infeasible to maybe so there's perhaps, depending on who you talk to, anywhere between 500 and 800 eucalypt species. And so it would be feasible to probably do them all on a DART chip reasonably cheaply once you've got it. And you do get a lot of, so I mean our simulations we've sort of done with 500 fragments or 1,000 fragments just to keep things going reasonably quickly, but really they're sort of getting, like I said, about 8,500 characters. And it does seem to have real potential for looking at hybridization because of the fact that it gives you this random snapshot over the whole genome. All right.
What's the depth of it? Sorry, how old are these guys? What are we looking at in terms of the? Because these are sort of random cuts, right?
So eucalypts, how old? I don't know if it would be something. Yeah, I'd just be guessing. If I had to guess, I'd probably say something like less than 50 million. But I mean, imagine maybe 10, 20. I don't know. Actually, erase that from the video record. Let it be known Barbara has no idea how old eucalypts are and forgot she was being videoed and shouldn't have opened her mouth. Long later.
Any other questions? Thank you again.